Hello everyone, and thank you for joining this webinar today. I'm Andrea Gramtzliuk, Head of Process Analytics Development here at Sartorius BIA, and with me it's also Tristan Kovacic, a leading scientist in my team, and together we will present how monolithic chromatography can improve safety of mRNA LNP therapeutics and vaccines. Our webinar is divided into two parts. First, I will introduce you to the monolithic columns, and then I will present you analytical methods for mRNA drug substance characterization. Tristan will introduce you to our novel analytical technology for drug product, and then he will expand into behavior of LNPs on monolithic columns. A few words about us, Sartorius BIA Separations. We are a leading developer of monolith technology and exclusive producer of convective interaction media, monolithic chromatographic columns. For more than 20 years, we offer these monolithic columns for purification as well as analytics for gene therapy and vaccine markets. We are based in Slovenia, western part of Slovenia, border to Italy, where we have world-class facilities for manufacturing of these monolithic columns, for research and development, and for contract development activities. Our key applications are adeno-associated virus, adenovirus, plasmid DNA, mRNA, LNPs, so all large biomolecules. So in 2022, we also joined Sartorius family and become Center of Excellence in Gene Therapies. This is our comprehensive product portfolio. It is divided into three parts. First, we have simultus, which are preparative columns. So they are available in different formats from 1 ml to 40 liters. They are ready to use, no pre-packing is required, and they are coming in different chemistries for different applications. So then we have screening chromatography, so where we are using for small volumes, we are having CMYC discs. And then for high throughput purification, we offer 96 valve plates. They're coming in, again, both products, they're coming in different chemistries for different applications. We have then also analytical columns, which are called our product. It's CMAC. They are specifically designed for high resolution and high reproducibility analytics. So they're specialized again for different applications. We have like plasmid DNA for different isomer separation. We have for empty and full separation. Now we have QRHR column and so on. So with the columns, we also provide our analytical platform, which is called PATFIX, and it can be used for in-process control and product release quality control. In addition of all these products, we also offer biomanufacturing development services, and this is that for process development, for analytics, and immobilization services. So now I will provide you a bit more details about the monolithic columns, their characteristics, and how they perform in comparison also to the porous particle media. So on the right side of your screen, you have the image of monolithic columns. So they are produced as a single piece of material. They are pre-packed and ready to use, comparing to the porous media where packing is required. So on monolithic columns, they are structured in a way they offer convective mass transport, so no diffusion, so which is much faster process comparing to diffusion limitation, which we have with porous media. So monolithic columns, they have channels. So we have all these network of interconnected channels. So there is no dead ends, so which is resulting in no entrapment of these large biomolecules. Comparing to the porous media, when quite often these large biomolecules are entrapped into the pores. So the whole surface of channels is instantaneously accessible to large biomolecules, which is resulted in high binding capacity for large biomolecules, comparing to the porous media where we have lower binding capacity. So size of these channels, we have they are from 1 micron to 6 micron diameters, which is really enables us much faster flow rates and lower back pressure, comparing to porous media where we have with high back pressure, so slower flow rates are needed. So those are one of the reasons why we recommend or monolithic columns are really applicable for large biomolecules. So we know that for proper characterization of mRNA during development and also during production, really it's important. So that is specifically to ensure drug quality, efficacy, and safety. So USP release the whole quality parameters for characterization and a release of mRNA drug substance. We are going from identity, content, integrity, potency, safety, and other. Today, I will focus more on purity quality parameter, where really we developed process analytics to monitor product-related as well as process-related impurities. So above we have the workflow for mRNA drug substance production. It starts with linear plasmid DNA, which really which serves for as a template for IVT reaction IVT reaction production. So then we go through different process or purification processes. First is capture process, and then it's polishing process through different dye filtration processes until before we are getting pure mRNA. So the whole production workflow is then supported with analytics. So specifically for IVT reaction monitoring, we are using Prima-S analytics. I'm going to talk into more next slide until we get to the integrity assessment of the mRNA product. We are using SDVB analytics. So this is chromatogram on the we have a UV chromatogram of IVT monitoring using Prima-S column. The method really separates and quantify all these key reaction components like capping reagents, nucleotides, all these NTPs, DNA template, as well as we are monitoring mRNA. So this method, it's really enables us optimization of IVT reaction monitoring. It's applicable for all RNA modalities, and now it's also been using also for quantification of process-related impurities, specifically for capping reagents and for NTPs. The same analytical method we are also using for determination of capping efficiency. We adapted method which was published by Chiron and all in 2017. So mRNA sample is digested with these two enzymes, and as we see, the uncapped mRNA is digested into nucleotides. However, the capped mRNA stays intact, and then quantification of the mRNA is been then monitored determined by Primias analytics. So this is one of the fastest analytical methods to determine capping efficiency in current field. So then we have for determination of poly-A tail, we developed ion pair reverse phase lipid chromatography, where we are using only UV detector. So mRNA sample is first digested with RNase T1. So RNase T1 cleaves after every G residue in 5 to 3 direction. So during this digestion, poly-A tail stays intact, and mRNA is degraded into shorter fragments. Sample is then purified by affinity oligodetic column, where we capture poly-A tail fraction, and this poly-A tail fraction is then analyzed by ion pair reverse phase chromatography. So this is like typical UV chromatogram. And the poly-A tail length is then analyzed by commercially determined by the commercially available letters. Ion pair reverse phase chromatography, it's also used for detection or determination of impurities. So here we have a separation of mRNA species according by hydrophobicity. So shorter mRNA fragments or even NTPs are eluting prior the main peak, and longer fragments, also like double-strand RNA, but other longomers are eluting after the main peak. So this is one of the methods which is primarily used for determination of impurities. And the same method it's been also been applied for detection or for determination of mRNA integrity. So here we are using the golden standard. Really, it's capillary gel electrophoresis. However, we developed the method where we are using a reverse phase column with ion pair conditions. This is on the top. We have UV chromatogram at 260, where we see for mRNA peak, we see some kind of front peak and the main intact peak. So then we are using for fragment determination, we are using first derivative of UV260 signal, where is the maximum of this first derivative. There is a split peak, and the frontage represents fragments of mRNA, and then the main peak is really like intact peak. We also compare these results with a capillary gel electrophoresis, and as you can see, very, very comparable results are obtained. So more details about the methods, it's been also published in the listed paper. So now I will pass the word to Tristan. So Tristan will present you the novel technology for monitoring and determination of crucial LNP parameters like encapsulation efficiency, size and size distribution, and mRNA integrity.
Okay, thank you, Andrea. Welcome from my side as well. So today I will focus on the drug product of this drug substance. So here we are talking about mRNA LNPs. And what we know now is that most of the time the analytical methods for LNPs are kind of poor predictors of in vivo behavior. There's many aspects that we put into a system like this when we introduce lipids. First of all, is just the lipid component itself. We have the purity, we have the assay and so on of the lipids that we put in. This is just the first step. Then the next step is the formation of this kind of a particle, where we get more of a morphological question of how this forms, how well it forms, how stable it is, and so on. So once we go here from drug substance to drug product, we introduce a lot of new variables and questions that we need to answer. Now, for particles like this, normally we need some extensive sample pretreatment to analyze them, or we need some labeling methods to ensure that we observe the things that we want to observe. There is a limited understanding of morphology of such particles, which then makes it harder to analyze them and to know how to manipulate them. We have certain presences of empty particles there that ideally we would want to see. And then we also have certain mRNA-related impurities that are forming during this encapsulation and then storage process of LNPs. They are inherently unstable just because they are a kind of a calculation of all intermolecular forces just held together. There is no covalent bonds or things like that. And they are quite diverse and heterogeneous just based on the fact that many different lipids are used in certain different formulations, which makes it also kind of hard to generalize the characterization process. And all of these things can lead to unsafe and unpredictable products if we are not addressing them in the development stage. Now, we at BIA took this challenge and we wanted to make a system, a chromatographic system to analyze certain aspects of LNPs and to make sure that we don't need any sample pretreatment or any labeling methods. Now, for this setup, we built a two-dimensional chromatographic system, utilizing monolithic columns, because monolithic columns are suitable for large biomolecules. They give good recovery of them, and you can achieve good separations by utilizing different column chemistries. Now, this is a simplified version of this setup, but basically we are starting with an LNP sample and we are diluting it into an appropriate buffer and loading it onto the OH column. An OH column is a hydrophobic interaction column. It binds very strongly particles, especially hydrophobic particles, and the larger they are, the stronger it binds them. Now, under certain conditions, this then enables that LNPs bind very strongly, but the free RNA does not bind to this column. And thus is the first step of this process. So we are separating the LNPs from the free RNA. Now this free RNA gets passed through and it gets to the second column, which is the SDVB column. With inline diluting switching systems, like a switching valve, we get the stream coming from the OH column, inline diluting with appropriate buffers and loading onto the SDVB. And here the free RNA is loaded and is able to be characterized. Now this SDVB column is the one Andrea was describing before. This is a reverse phase. Here we are operating it in a reverse phase ion pair mode, which enables binding of RNA, separation by size of RNA, integrity of RNA, and things like that. So here then we bind the free RNA and then we elute it with increasing acetonitrile to characterize it and quantify it by UV vis detector. In the second step, during this time we are keeping on the OH column the LNPs, and then we are eluting them down from the OH column and binding them onto the SDVB column. In the meantime, they also get past the multi-angle light scattering detector, which enables detection of particles, but also can enable you to calculate the size of these particles. So let me move on to the next slide to present to you this in a more detailed manner. So this is an animation, and here we have, for example, it's a sample with an LNP, with encapsulated RNA, and then with free RNA. Now this gets passed through, and the particles bind to the OH column, while the free RNA gets passed through, and inline diluting with the buffers for SDVB column, it gets bound to the SDVB column. And then, as I described before, analyzed on the UV-Vis detector. And here we quantify the free RNA part. Then, by reducing conductivity of the buffers, we elute LNPs from the OH column. We analyze them on the Miles detector, and then they get mixed with the appropriate buffers to bind onto the SDVB column. Now, what happens here is that we don't actually need some detergent treatment to open up particles, but we're utilizing kind of a solid state opening. And here the particles get, they're bound so strongly to the SDVB column, which is highly hydrophobic, that we cannot get intact particles down from the column. What we basically do is that we start increasing acetonitrile, and first we get out the free RNA. And free RNA gets eluted here and analyzed, and here we quantify the encapsulated RNA part, and we can compare it to the free RNA that we quantified before. Then, after a certain acetonitrile percentage, we also start eluting separate lipid components here. So it is also possible to add a detector like a charged aerosol detector or an evaporative light scattering detector to quantify and analyze those separate lipid components coming down from this stream. But otherwise, we are analyzing, in this case, the encapsulated RNA part, and we are quantifying it, and then we are also able to characterize certain impurities and its integrity as well. And this is how a typical chromatogram of this would look like. So we would get a free RNA part, the one that didn't bind to the OH column, and then we would get the encapsulated RNA part released from the particles on the SDVB column. And this would be determined by the UV 260 nanometer detector. And down here we have the multi-angle light scattering detector, which detects particles after they get eluted from the OH column. Now, here is a certain proof of the opening of particles, where if we put the multi-angle light scattering detector behind the UV, we observe no signal in the multi-angle light scattering detector. We believe that the sum of all the hydrophobic forces with an intact particle is so strong that basically the column, the SDVB column flattens it out, and then we elute each separate component down from that column. Now, the first parameter, the most obvious one that we can determine from an analysis like this, is the encapsulation efficiency. Encapsulation efficiency nowadays commonly done by the ribogreen fluorescent assay method is a crucial parameter, is a critical quality attribute of LNPs because we want to know how much RNA within our sample is actually can transfect into the cells by being properly encapsulated. Now, this is an orthogonal method to the widespread ribogreen assay. It also gives you quantification, total quantification of RNA from the UV detector, from a calibration curve of your RNA, but it also gives you this ratio between the free RNA and the encapsulated RNA. And this is obviously a slightly different way of separating the free and the encapsulated part than on the ribogreen assay. Thus, we say it is an orthogonal method to that. But it definitely gives you that value, and then you can decide whether or which method you think it's best at separating those. But there is another critical question is what to do if you are encapsulating multiple cargos within your LNP. Now here we have a case like this where we have a RNA of size of 1,000 nucleotides and an RNA of size of 4,000 nucleotides. Now in a chromatogram like this, we have the free RNA part and we have the encapsulated RNA part. And in both of them, we can observe both RNA species. And we are able to determine the encapsulation efficiency for both of them separately because the SDVB column is able to separate them. Now, in this case, we see that they are quite close together, although still separated. Then this very much depends on what size your RNA is. But this method can be easily tuned with a gradient with certain buffer compositions that you can achieve the selectivity that you want to. If you have like other combinations of RNA, for example, if you're using for gene editing, you're using a CRISPR technology with Cas9 and the guide RNA, those are much easier to separate because a larger difference in size. But this can be easily done, and you can have a separate each encapsulation efficiency value for each of those. So you have much more information using this. Then in the multi-angle light scattering detector, you can also observe sort of the particles that come down from the column. And the OH column is able to separate particles by size. I will talk more on that later. But then you can observe also how heterogeneous your samples are. And in this case, you can see we have a few peaks that show us the particle part of this sample, which gives us an information of a quite heterogeneous LNP sample. So another thing you can determine using this method on the UVVs detector with the SDVB column is the RNA integrity. Now, Andrea talked about this method before, which is the results are totally comparable with capillary electrophoresis. But in this case, what the powerful thing that we have is you can, before you'd actually do the integrity analysis, you'd separate the free and the encapsulated RNA. Now, if your sample is 100% encapsulated, this might not matter, but you will still need to do some manipulation of the sample with a detergent or so to then inject it onto the capillary electrophoresis to determine this integrity. But here you inject the sample directly onto the SDVB column, and you get separate values of integrity for the free mRNA part and for the encapsulated mRNA part. If your sample is well encapsulated, ideally you should have a very good integrity for your encapsulated part, and you should have a worse integrity for your free part because it's less stable in that environment, especially if you are then storing this sample maybe at above a freezing temperature if you store it in a fridge or so. And here we can see on this chromatogram, this is the free RNA part, and this was a sample that was aged for 4 days at 35 degrees. And we can see starting here the red line at day 0, it was quite intact. And then after heating for some time, this line moved left and left, and it broadened out the peak, which means that we are getting a larger percentage of the fragmented RNA. Now here, for the encapsulated RNA part, this is not so obvious, which means that the RNA is more protected towards degradation once it is encapsulated. And these are more specific numbers. So for example, for the free RNA part in this sample, we went from 29% fragmented to 69% fragmented to 79 just in a matter of 4 days. But this was hitting at 35 degrees. And whilst at the same time, the encapsulated part went from 15% to 19% to 30%. So increasing, but much slower than for the free RNA part. Now, another important thing is the RNA impurities. Now, once we start introducing lipids, we get another variable in there that could cause some mRNA related impurities that can be just maybe less active, which might be less of a problem, but it can also maybe cause some other problems in your products. Now here I am showing, this is a thing known that was first published by Moderna a few years ago, but more recently by Kashiba in 2024. I'm showing here the formation of aldehydes from the ionizable lipid. The ionizable lipid is a tertiary amine, and there is an oxidative pathway for amines like this to form aldehydes. Now, a tertiary amine like this can be oxidized to an N oxide and then further hydrolyzed down to a secondary amine and an aldehyde. Now this aldehyde obviously is very reactive, very electrophilic, and is known in the body as being able to alkylate nucleic acids. And an aldehyde like this can react with your nucleobases and form adducts like this here, which obviously drastically changes its chemical properties and properties of the RNA as a whole. Here we can see some sort of graph how the activity is reduced after storing at 4 degrees at the adducts during which these kind of an adducts are forming. Now, in the LNP switcher, the two-dimensional setup that I was describing before, we can observe those impurities because they are we can separate. We are here basically separating RNA species by hydrophobicity. Now, if you have pure RNA, this means, and you are using a ion pairing reagent, which we are a triethylamine in this case. Basically, the larger RNA it is, the stronger it's going to bind to the column because there's more of these triethylamine lipid tails that will bind to your column and increase these hydrophobic interactions. Now, once you start forming impurities like this that I was showing here, this is a much stronger hydrophobic tail than just the triethylamine. This will make it stick to the column much stronger. And what this means is that even though you might not get a very large difference in size of your RNA, which would be observed by capillary electrophoresis, you will get a very strong change in hydrophobicity. And this enables then quantification of such impurities on a reverse phase column, especially here in this case a reverse phase monolithic column because it enables binding of large species like this RNA. So once we open up the particles on the SDVP column, we are then eluting first here the kind of a pure encapsulated RNA part, but then later at a later percentage of acetonitrile we get down this part, which is it's a mixture of RNAs of different levels of lipidation, but it is a much more hydrophobic species. And this is obviously highly dependent on your lipid choice, on your storage conditions, and on your raw material purity. Because if you're introducing this purity even from the start, this is going to happen from the start. You're going to observe a lot of this. You can also form this during storage, and the higher the temperature is, the more of this you're going to form, just because there is going to be a slow degradation, an oxidative degradation of that part of that species, as I described before. But it is also very important um, and crucial when you're designing lipids to take care of this, because some lipids are going to be much more prone to degradation like this than others, just based on their chemical properties. Now here, this is shown from an article by Packer in 2021. This graph here, where we see that relative protein expression is CE is not very well correlated. So RNA integrity determined by capillary electrophoresis. You basically have from 80 to 100 ranging values, but the protein expression is much more heterogeneous here, ranging from 40 to 100%. Now, if you're analyzing it using RPIP, which is a case like this, you see that the more pure, the higher integrity the RNA has, the higher the relative protein expression is going to be, because you are taking into account here these impurities that you are not separating by CE. So it's important. Very important to do RPIP analytics as well. Now, to move on a little bit from the RNA part of this analysis, I'm going to talk briefly about the LNP size and size distribution measured by the LNP switcher method. Now, LNP is size is a crucial parameter. It enables LNPs to enter cells, evoke an immune response, and evade clearing mechanisms. Ideally, we want a uniform immune response, so we want a uniform size of LNPs that we are forming. Different techniques are used for this: NTA, DLS, cryo-EM. Now, techniques like DLS have a problem that cannot separate populations very well, um, and you basically can have a very large response of large particles that will overshadow a lot of the small particles that you have. So ideally, you want to separate those particles a little bit just to make sure you're not overlapping things. And it is LNP switcher method. We are eluting LNPs down from the OH column, which is a hydrophobic interaction column, and is separating particles by size, just because the larger particles are going to have a stronger hydrophobic interaction, like a sum of all their forces, and they will elute later from the column. Now, a multi-angle light scattering detector is able, we are here recording 9 angles, and these 9 angles, from a ratio of these 9 angles, we are able to determine the size of those particles. And we also observe, plotting like this pink line here. This is the size is getting higher the later it elutes down from the column. But comparing this to other techniques like NTA or DLS, using a Berry mathematical model, we have a good relationship between those two values, and we can determine relatively reliably the size of the LNPs coming down from the OH column. Now, to talk a little bit more about that kind of the interactions, I'm here describing the OH column interactions with the LNPs. Now, this is now a chromatogram of just a separate setup, which is a one-dimensional setup, where we are binding the LNPs onto an OH column. And in a setup like this, we would have free RNA going into the flow through, and we would have the LNPs then eluting down from the OH column after we get to a certain low percentage of the loading buffer. Now, this enables obviously separation of the free RNA and of the LNPs, but with the multi-angle light scattering detector, we can also observe some of these larger fractions that we might want to get rid of and that have very little RNA in them, or basically no RNA in them. Now, binding and eluting LNPs to the OH column works like this. So if we scale this up a little bit, we get a more preparative chromatogram like this. And we can collect these fractions that we bind onto the OH column, and then we elute down and collect the particles. Now this here we are showing at the NTA analysis, nanotracking analysis of size of LNPs before the OH column, and after we collected this peak from the OH column. Now we see that the size is very similar; they remain largely intact. And now if you would separate certain fractions, then you would get different sizes down from there. But if you're collecting the whole thing, basically your size remains intact and the integrity of the particles remains intact. Now, if we're talking about this in an analytical way, we can also talk about scaling this up. monolithic columns are very good at being able to scale up. You have kind of flow independent properties, and we can basically straight after formulation of LNPs, which we would call here the crude LNP formulation. Now without dialysis or TFF, we would then inject that sample onto an OH column and then collect it down from the OH column by reducing the loading buffer. And we would collect those particles. And we can see that then the pure, which would be collected from the OH, would have a higher encapsulation efficiency just because we are getting rid of that free RNA part. And from the multi-angle light scattering detector, we can also see the concentration of particles, which is this is like the same amount injected. Orange line compared to the black line, we are increasing concentration of those particles by quite a lot. And a very big thing is to have we have a very good RNA like encapsulated RNA recovery, using these going straight from the formulation towards then this sort of purification. And we are increasing, for example, encapsulation efficiency, but the total RNA content is important here, where we are only losing 5% of the total RNA that we are loading onto the column. Now this going forward from there would enable a certain workflow like we have here. So for those of you who are familiar with encapsulation part, here we would mix RNA in an acidic buffer with lipids in ethanol. We would form the LNP-mRNA in an acidic, that would come out in an acidic solution. Now for us, we would then dilute and neutralize this to get them to a pH 7.4, and we would have this sort of a sample that we would then load onto the OH column by inline diluting with the appropriate loading buffer. And in the flow through, we would then collect the free RNA, which can also be regenerated if you want, depends on how low your encapsulation efficiency is. And then once we have LNPs bound on here, we can then elute them down, and then get them pure and concentrated LNP RNA formulation, which is also then just be able to be sterile filtrated and stored for any further use. Now, this is now sort of switching things a little bit to a lipid composition analysis, which is again a crucial part of the RNA critical quality LNP-mRNA. We are introducing lipids, and we are certainly introducing normally 4 different components. So the ionizable lipid, the phospholipids, the cholesterol, and the pegylated lipid. Now those 4 lipids, we want to be able to separate them on the chromatographic column and to quantify them. Now, in addition to quantifying them, you can also determine basically how much you're losing and what other impurities they have before, and what other impurities they might be forming during the storage and encapsulation part. Now, if we have a C4HLD column using here, this is another one of our monolithic columns. This is can be used in both hydrophobic interaction mode or reverse phase mode. Now here we're loading the LNP straight. So the formulation we are using, we don't do any extraction of lipids before, but we are just injecting straight the LNP onto the monolithic column. And this, again, because C4 is highly hydrophobic, it works in a kind of similar mode to SDVB. We cannot get intact particles down from there, but we can, by increasing organic solvent, we can then elute each separate component down from that column. And here, if we utilize both the UV detector for RNA and then another sort of general detector for lipid detection, we can observe both, so the RNA and the lipids. Now in this case, we have an ELSD chromatogram, evaporative light scattering. We can also use charged aerosol detection. This basically, we need a general detector that is able to detect small molecules that have no chromophores, because these lipids have no apparent chromophores that could be then monitored by the UV detector, or at least their signal is not high enough to do that. But we are able to basically inject the intact LNP on here and then analyze each separate lipid separately. And what this gives us is also the actual lipid composition that we are getting with these lipids. And we are certainly at the start with formulation, we are putting in a certain mass ratio of lipids, but then probably the actual lipid composition within those is not the same that we added to the start. Some might get lost to stick somewhere, or then especially this becomes important if you're doing storage stability screenings. And you might want to see how well you retain your lipids and during certain storage conditions. So we, for example, in this case, you can see from the initial lipid mix in the kind of a crude LNP after formulation, we would see that some of the ratios of these lipids changed, not dramatically, but maybe important enough for some of your applications. This also has been recently published by us, so you can also check this for more details on how to use this method. So thank you from my part. This has been it's been nice to present you this, and now I want to pass the word to Andrea to make this some certain conclusions.
Okay, so thank you, Tristan. So just I would like to recapture or summarize, our webinar, what we presented. So really what we demonstrated is that we provide you with LNP analytical platform, which really enables comprehensive drug product characterization. So with that kind of analytics or platform, you have direct injection of LNPs. So simultaneously particle opening on the column. So, and with this type of analytics, you'll be able to determine crucial LNP parameters like determination of encapsulation efficiency, also co-encapsulated cargo quantifications, size and size distribution determination, and on the same time you'll be able in one analytical assay also determine the mRNA integrity for free as well as for encapsulated mRNAs. Also, with that type of analytics, you'll be able to monitor mRNA-related impurity quantification as well as lipid component separation. So with that, I would like to thank you, first of all, thank you, our managing director, to enable us to do this work, my team, which is also on this photo, and also colleagues from different departments. So now we would like really to thank you to listening this webinar, and we are happy to taking some questions. And yeah, let's put our camera on. So as we see, we received already some questions, and I will just start and pass it primarily to you, Tristan, because it's more related to our novel technology. Specifically, let's start with this one. If LNP switcher method is applicable also to other lipid nanoparticles like liposomes, emulsions, and so on.
Yeah, hi. Thanks, Andrea. So yes, this can all be applicable to other particles. Obviously, the certain conditions would need to be changed because some particles have different properties of like they can be more stable, like harder to open. But we have tested things like liposomes before, and we are able to quantify those as well. It's just for it just needs to be optimized a little bit, but it is possible and has been done before.
Yeah, thank you. Could OH column be used also for LNP purification, buffer exchange in production?
Yes, this is maybe a little bit of what I was alluding to before with those certain slides I had on the on now using the OH column for kind of scaling up that analytical process. But yes, you can get rid of ethanol and certain buffers from within your formulation sample and then change it to another buffer using the OH column because whilst loading on the column, you get rid of those things and you concentrate the particles.
Okay, thank you. And relating to OH column, is OH column for column also possible in GMP environment?
Yes, PIA, I
Yeah,
believe, is producing
yeah.
the OH column in many different sizes in cGMP format as well, and different pore sizes as well.
Another one interesting one is if with this novel technology, can we differentiate between full and empty LNPs?
Yeah, a very good question. Now, a straightforward answer for a direct setup like this would be no, but there are certain things we observe while separating on the OH column that some fractions have little to no RNA. But there is also other applications that we are working on with other column chemistries that we believe are able to do that. So, we will keep you posted on that.
Another one is more related to size of nucleic acids. What size of nucleic acids can be analyzed using this analytical setup?
So we've gone as far up as the saRNA size, so it's about 10,000 or so, or even more of the size, and we have no problems there. Like even the larger the RNA, the better the monoliths are to analyze them and purify them. So in terms of RNA size, there is really no limit here. Maybe just keep in mind that whilst having a very large RNA, you also change drastically the properties of LNPs. So then, if the LNPs are much larger, that maybe can become an issue, but you don't want to have them that large anyway. And here we are using on the OH column 6 micrometer pores, so up to like a few hundred nanometers, there is really no problem for the LNPs as well.
And another one, can you detect degradation products in LNP formulations? This was partially already addressed during your talk.
Yeah, so yeah, if we're talking about
Yeah.
RNA degradation products, then that's yeah, we've shown this from the integrity part and then the RNA lipid impurity adduct sorts. Now also others on the SDVB, we're separating also other species, so like double strand RNA, things like that. If we are talking about lipid impurities, just straight small molecule impurities stemming from the lipids, that can also be detected using the method that I described at the end if the threshold is high enough.
I will answer also that one. So, OH columns works for hydrophobic interaction. As you mentioned, does it specifically binds with LNPs or also the free lipoids, if any?
Ah, yes. So if you mean yeah, like empty particles,
Yeah.
it depends on their size and their hydrophobicity. If they are more hydrophobic than the full particles, which we believe they are, and they're also less stable, then you will have stronger binding to the OH column. But in principle, if they are of the same size and the same like lipid composition, they will bind the same.
So I think that this is it for now. So all the rest will answer and will address via emails individually. So really, thank you for both of us, from both of us, to listening this webinar, and hopefully it was, yeah, useful. So, and we
Yeah.
shared some important information.
Yeah, thank you very much. Thank you for your questions, and yeah,
Thank you.
email us if you have any others or any other
Yeah.
wishes. Yeah,
Thank
thank
you.
you.